So thank you so much, and I'm incredibly excited to be here today and to join with you guys in efforts to develop better treatments and cures for cystic fibrosis. And in particular, I'm going to be telling you about bacteriophages, which I think are a frontier not only in cystic fibrosis research and lung infections, but also in medicine in general. And I'm going to be telling you two stories about one phage or one set of phages that I think we can use to help us fight infections in cystic fibrosis, and then about another phage that I think actually causes disease in cystic fibrosis lung infections. So two stories for the price of one. And let's see here. And so viruses like bacteriophages have evolved over the years to adapt to their particular prey species. So individual strains of Pseudomonas have their own phages. Staphylococcus, which is another lung infection in cystic fibrosis that's important, has its own phages. And that's going to be important later on in the talk. And then these are abundant at sites of bacterial colonization and infection, like the cystic fibrosis lung. The other thing to know about bacteriophages is that they come basically in two flavors. One are lytic phages. And if anybody in this room has ever had, influenza, and I imagine most of us have, this will be very familiar. The viruses get into the bacterial cells and they replicate and ultimately cause problems for the cells that they replicate in. But bacteriophages have another trick. There's a subset of bacteriophages that actually live in the genomes of the bacteria that produce them. They're called lysogenic phages, and they actually partner with the bacteria that they inhabit to do all kinds of dastardly things in the context of lung infections. And again, that'll be important later on in the talk. And so really, I've got two stories to tell you, as I mentioned earlier. The first one is about how we can use lytic bacteriophages to fight lung infections in CF. The second one is about how a particular lysogenic phage actually causes disease in cystic fibrosis. And this is a story that we've been working on for a number of years with my good friend and colleague right over there, Dr. Carlos Mila. Okay, the bacteria that we're going to be talking about here is Pseudomonas aeruginosa. And I think most of the people in this room are familiar with this bug. It's obviously very problematic in cystic fibrosis and in other contexts. It's what's called a gram-negative bacteria. So it's not a virus, it's a bacterial pathogen, and it causes skin and lung infections. And the reason why it's so problematic is because of antimicrobial resistance. When I'm on clinical service, I'm an infectious disease doc, I routinely come across versions of Pseudomonas that are resistant to pretty much any antibiotic that we could ever throw at them. So this is a big problem in CF and in other contexts. And so the first story that I'm going to tell you with these lytic phages are these guys here. They kind of look like lunar landers, and they'll be very familiar to many of you folks from the perspective of that being a prototypical virus shape. And as you guys may know, there's been incredible progress just in our lifetimes over in terms of cystic fibrosis care and outcomes due to inhaled antibiotics and some of the modulator therapies that we'll be hearing about later. But the problem is as we're learning, these agents have not fully taken care of the problem. This is a paper from Pradeep Singh's group at the University of Washington that came out earlier on this year, showing there on the left that sputum production is way down with modulator therapy over time. So the more months you're on this, that improves. But actually, bacterial colonization with Staphylococcus and Pseudomonas doesn't in fact go away. I don't think any of us really knows what that means from the perspective of long-term outcomes. But it's safe to say that infections are likely to be a consistent problem in CF, even with these new drugs. And then when you think about that, the bacterial pathogens that are causing these problems over that longer period of time, you're going to see more antimicrobial resistance. And this is just some data from the CFF annual report from maybe a year or two ago that looks at antimicrobial resistance in Pseudomonas and its prevalence in the CF community. And as you can see, this is also a problem that's not going away. So even with these new drugs and thinking about where we are with this disease, we're going to need better ways to tackle antimicrobial resistant Pseudomonas and Staphylococcus. How are we going to do this? And I'm going to argue that in looking forward to thinking about this problem, we can actually look back to this guy here. Félix de Herelle was a French microbiologist who made a discovery in 1917. He looked at a petri dish, very much like this one here, and he saw sort of holes in the bacterial culture. And he's the one who named these things bacteriophages. And what that means, bacteria is obviously a bacteria, and phago comes from the Greek word for eat. And so he called these bacteria eaters, as I showed you a moment ago. And he's really the person who kind of discovered bacteriophages. And we don't need to dwell on this entire timeline. But the point with it is there were some successful trials of this for infections, in particular for cholera. The data there on the right is original data from one of the first phage therapy trials in 1931, which is kind of fun. Anytime you can look at 100-year-old data and it's relevant today, that's kind of exciting. But the problem is when he tried and others tried to commercialize phage therapy over the subsequent decades, it was really difficult. The biology, as I mentioned earlier, is complicated. There are lytic phages and lysogenic phages. Purifying these things wasn't something they could do. And they had some problems that I think we're just starting to be able to address today from the perspective of host range and things like this that we'll talk about. The other thing is conventional antibiotics really took off starting in the 1940s, as many of you guys know, and that sort of moved phage therapy to the sidelines of antimicrobial medicine. But I'm going to argue that phages are relevant again today, and that's because of this. Antimicrobial resistance, as many of you guys know, is an enormous problem, and not just in cystic fibrosis, in many, many contexts. The estimates are that currently 700,000 people a year die of antimicrobial resistant infections, and the bill for this is over $100 trillion a year. Of course, the bill is even greater when you think about human suffering and just the challenges that this poses for health in our patients. But if you think about this moving forward, this is only going to be a bigger problem. The UN estimates that by 2050, 10 million people a year will die of antimicrobial infections. So we really need to solve this, both for our CF patients and for the world. And so the question is, getting back to Félix de Herelle, is there a role for phage therapy? Now, we have a speaker later on, I think tomorrow, Dr. Aslam, who's going to go into this and the history more. And so I didn't want to cover all of phage therapy as it relates to cystic fibrosis, because I'm going to imagine she's going to talk a bunch about that. And instead, I'm going to talk about something a little bit more specific. But I wanted to kind of highlight some of the advantages of phage therapy and some of the issues that we're trying to tackle in the field. The advantages are that phages can amplify themselves in the setting of bacterial infection, and then they're exquisitely host-specific. So if anybody in this room has ever gotten a urinary tract infection after taking antibiotics, that really won't happen with phages because they target just the bacterial species that they're after and not all of the bacteria in your body. That's kind of attractive. They're effective against biofilms. And as many of you guys know, in cystic fibrosis and in other settings, bacteria essentially align the airways, and that makes them very difficult to clear. Phages are also non-toxic. They don't get into human cells. And so problems like deafness and kidney issues that we run into with our patients on antibiotics really are much less of an issue. And then they're inexpensive. We think that these are therapies that the world can benefit from. But they have some disadvantages. That narrow host range becomes difficult in the setting of CF, for example, where some of our patients can have hundreds of individual Pseudomonas isolates in their lung. And so how you cover each of those individually with phages, as I'll talk about in a minute, is a big problem. There are also issues around storing these things and turning them into drugs that we're still trying to figure out. And again, I think Dr. Aslam will talk more about this tomorrow, so I don't want to get too much into that space. But what's interesting is that, you know, I think many of us in the field are inspired to try to use phage therapy as an approach for chronic bacterial infections from cases like this one. In 2017, Tom Patterson was one of the first patients in the United States to successfully receive phage therapy. I think Diane Smith will, in terms of her daughter Mallory, had an experience that she'll share with us. But the point with it is that this case here showed us what the potential for this approach can be. Mr. Patterson had a pancreatic cyst and ended up with a multi-drug resistant Acinetobacter baumannii infection. And he was critically ill in the ICU, but he got phage therapy that saved his life. And there have been other cases. So in the news, maybe a couple of years ago, was a case of non-tuberculous mycobacterial infection that happened in a CF patient, a 15-year-old girl who got phage therapy with engineered phages that cured her of her mycobacterial infection and allowed her to live for a number of more years. And these two patients are not alone. This is a paper we put out a little bit ago looking at the number of successful cases as of 2020. I think there's even more now. But the point with it is that there's this whole little industry that's sprung up over the past few years, and probably a couple of dozen companies that are trying to develop phage therapy for our patients. And when you look at these successes, and I think in the United States, we're up to several dozen now. Around the world, it's probably more than a few hundred. And when you look at these, I mean, it's incredibly exciting, although I will say that there are a lot of unsuccessful cases that don't really make it into the literature. The problem, though, is when you get to clinical trials, it's been incredibly difficult to scale up what ends up being sort of a artisanal, personalized medicine into something that all of our patients can use. It's also very difficult to scale these things up into therapeutics that, quite frankly, people can make money off of. Because if you're developing phage therapy for each individual patient and it ends up being incredibly intensive and involved, that's a difficult thing to build a business model around. Insurance companies don't reimburse for this. And so how we can develop this into a successful pipeline in CF and in other settings has been really challenging. A lot of the phage therapy cases that have tried to do clinical trials have in fact not been successful because adapting phage therapy into a kind of one-size-fits-all program has been difficult to do. And for this point, there are no phage therapy products that are approved currently in the United States. There are some for food use and for doing things like killing listeria that can get into cheese or salads or what have you. The FDA has approved that. But for human use, we have not seen this happen yet, again, because the clinical trial data has been lacking. So what can we do about this? So there are clinical trials that are underway. There's at least 24. If you go to clinicaltrials.gov, phage therapy clinical trials, many of them are in cystic fibrosis. They're supported by the CF Society and by other groups, the NIH. This is one that just ended prematurely because the company that was doing this, I think, ultimately ran out of funds. But this was done at Yale using a cocktail of 3 phages here that people received for 7 days. And what they found, these were all individuals who were on modulator therapy. And when they actually went back and looked, even though the isolates that they were targeting of Pseudomonas were eradicated or gone, they believe, the overall burden of bacteria in the lung, including of Pseudomonas, didn't necessarily go down, which was the outcome that they were looking at. And they're still analyzing these data, and it's only 4 patients with CF that got treated with phage and then 4 controls. But it's a little bit sobering because it suggests that if you look at the lungs of our patients with CF, that they have very many isolates that potentially would need to be knocked down and treated. And it's a little bit like whack-a-mole. You knock down one clinical isolate and then another one grows up. So how are we going to do this? How are we going to get rid of infections like these? Presumably in the non-tuberculous mycobacteria setting, there may be less diversity in all of this, but for Staphylococcus and Pseudomonas, we need better solutions. And to put this another way, we don't know how to generate phage therapy cocktails. So the 2 questions that we've been asking in the lab, or 2 of the questions we've been asking, are how can we expand the host range of phages to do a better job of encompassing that diversity? How can we whack a lot of moles instead of just one? And then how can we use phages and antibiotics together? And to sort of show you a little bit, maybe graphically, what this problem looks like, if you imagine each of those dots there is a bacterial species, and the phages that kind of mirror it are the ones that are sensitive to it. I realize the graphics look a little bleached out here, but the point with it is if you have a bunch of these different isolates and each individual person has their own set, it becomes very difficult to apply a generalized approach to this that, again, isn't, personalized medicine. And so the company that worked with the Yale group is this one here called Felix. And they developed a phage therapy cocktail that I alluded to a moment ago when I showed you their clinicaltrial.gov page. And it's called the PYT1 cocktail. And it covers, per their analysis, about 60% of Pseudomonas isolates. And apparently that wasn't enough. And so we approached this with a little bit of a different hypothesis. Our goal was to see if we could, by targeting the receptors, by determining the host range of the different bacteriophages, come up with a rational and sort of systematic approach to building these cocktails. And so what we knew is that essentially there are only a limited number of targets that bacteriophages can have on the surface of bacteria. They're things like the type 4 pilus, lipopolysaccharide or LPS, and flagella. Flagella are these big hairs that Pseudomonas and other bacteria use to swim around in the lung at other contexts. And so what we thought, or what we hypothesized, is by combining phages that use different receptors that we could come up with phage therapy cocktails with broad host range. And this is a little bit like with HIV. I think many of us in the room are old enough to remember when there was one HIV drug and everybody essentially developed resistance to it almost immediately. But what solved the problem was coming up with cocktails of 3 or 4 antiretroviral drugs that together prevent the emergence of resistance by essentially attacking the HIV virus from multiple angles. And that's really what we were trying to do here. And so this is where the CFRI came into the game. Nina Penitzdorfer is a postdoc, or was a postdoc in my lab, who worked with Kevin Kim, another postdoc, to develop an approach which we call complementarity assays to identify what the receptors are for different bacteriophages. Because only if you know those can you then build a cocktail of phages that target different receptors. And essentially what you're looking at there on the y-axis is the growth of this bacteria. And we measure that through optical density and then looking at that over time. And I won't make you guys dig into too much microbiology here, but essentially what we see is that in the red line, some bacteria are immediately resistant to these phages, but most of them will eventually develop resistance over time. And what we can do is we can look at kind of the maximal growth, meaning if there's no phages there, and compare it against when there are phages, and come up with a number which we call the suppression ratio. And this allows us, having a suppression ratio index like this, allows us to quantify the amount of bang we're getting for our buck with these different phages. And you can see that there are different concentrations of one phage whose name is DMS3-Vir, and we see more suppression with more phage. That's really great. But what Nina did, which is kind of interesting, is she then went and took the bacterial isolates that emerge after phage therapy and put them, essentially under the same selection pressure again. And what she found is that sometimes resistance again emerges over time, but every now and again, there were bacterial isolates that were immediately and constitutively resistant to the phages in question. That's really cool, because what that meant to her, or what she hypothesized, was that this meant that they were throwing away the receptor that the bacteria used for, or that the phages used to get into the bacteria. Does that make sense? And so what those plots are there at the bottom, you see like something that looks like a, almost like a coffee stain. That's called a twitching assay. And so you can see how much the bacteria are moving around. And then the isolate that's now resistant to that phage, GMS3, isn't twitching at all. The reason for this is because it's essentially gotten rid of a structure called the type 4 pilus. That's the receptor for that bacteriophage and gives it that twitching morphology. And what Nina saw is that if you then go back and look at other phages, by developing resistance to DMS3-Vir, these bacteria were also resistant to other bacteriophages. That's really cool, because it means that there's a whole group of bacteriophages that are using the same receptors. If you then scale up this assay, what you can do is look across a whole bunch of different phages and identify shared patterns of receptor usage. And so what you're looking at here without going into all of what the different mutations are a group of phages that have combined or shared patterns of resistance either to LPS or to the type 4 pilus. And if you then scale this up even further to look at a whole bunch of different phages, you get maps like this one here. And so the first exposure and the second exposure refers to, this first screening round where we then identify resistant isolates, and then we come back and look at what they're sensitive to again. And you have a group of phages that rely on type 4 pilus, on the flagella, on oligosaccharides, and you get a map like this one, where you can identify what we're calling complementarity groups, right? And so this is almost like if you were going to try to reverse engineer the antiretroviral drugs in HIV and figure out what molecules and receptors they use, and then identify ways that you can combine them together to really suppress HIV resistance. Does that make sense? Okay. But where it really matters is in plots like this one. If we combine phages that have the same complementarity group, it turns out that we get less suppression. If we combine phages that use different receptors that belong to different complementarity groups, now we get really great suppression. And that's exciting because this gets you down the path towards actually developing cocktails that you can use broadly. And I'll show you more about that in a moment. Okay, the other question that you can use, these assays to answer is, can you combine phages antibiotics together in ways that give you more bang for your buck? And so there have been some studies on this. This is one of them from 2020. And what people have found is that it's a little bit idiosyncratic. Some phages work in a good way with antibiotics, and some phages work in a bad way. They actually antagonize the antibiotics. Just like different combinations of antibiotics sometimes work well and sometimes they get in each other's way. And the order in which you give these things matters, and whether you get synergy matters a lot as far as the outcomes with resistance. And so what Nina and Kevin did is then took that same grid approach and looked at the different families, the complementarity groups of phages, and compared them to classes of anti-pseudomonal antibiotics. And what we found is that particular complementarity groups work really well with some antibiotics and not others. And so what this begins to give you is a rational framework for combining not only different phages, but phages and antibiotics classes. Does that make sense? And so, I think I mentioned at the beginning that I'm an infectious disease doctor. And so when I work in the hospital, I have what's called an antibiogram. And basically, I walk around from patient room to patient room, thinking about the different infections they have, and I can look this up, and I can see which antibiotics, are associated with which level of resistance and how to combine these things. And so charts like this one give me a map, for essentially doing this now with anti-pseudomonal therapy. And we've now done this for staph. We're doing it for other organisms, and it's really cool because it gives us cocktails. So what does that actually look like? So here across the top are clinical isolates that Carlos over there collected in the CF clinic from our patients. Liz Bergener, who was a clinical fellow in our lab, then curated these things and put them together. I think at this point we have about 700 clinical isolates from different cystic fibrosis patients. And each of those numbers across the top is one patient's clinical isolate of Pseudomonas. Then on the side, you have different phages. Luz24 is a phage, POMCO01 is a phage, PAML L31 is a phage. And what you see is that they're, sometimes good, sometimes not. And that's just the way this looks. But if you combine them, they're mostly good. They work a lot better. If you then take astrianam, which is a common antibiotic that we use in our patients, astrianam, most of these isolates are resistant. And unfortunately, we see a lot of that in our clinic, and they don't work very well. But based on that matrix that I showed you a minute ago, you can then combine the astrianam with the phage cocktail, and now you're cooking with gas, so to speak. You've got really great suppression, and this is exciting. So let's supercharge this. Here, you're looking at 174 different patients' clinical isolates. And there on the left is the cocktail. Next to it is the astrianam by itself. And then there's that combination. And if you look there on the right, we made 3 cocktails like this. And each one of them by itself covers about 85% of the clinical isolates in this panel. when you combine them with astrianan, which again, most of them are already resistant to, or many of them are, these are longstanding CF patients. Now, these cocktails are working in 95% of patients, or more than that. So if you think of the trial that I showed you a moment ago that was done at Yale, where that cocktail covers anywhere from 50 to 60% of these cases, and you still got the emergence of resistance, this is exciting because by covering 95% of these clinical isolates, we're optimistic that we can do a better job of delivering phage therapy that's going to cover the Pseudomonas that's in our patients' lungs. Does that make sense? We also have data like this for, again, Staphylococcus aureus, and we've looked at this in the context of biofilms. And where this begins to get you, beyond having cocktails of therapy that can cover broad things, is actually, I hate to say it, but commercialization. Because once you have cocktails like this, you have a product. And once you have a product, you can get industry interested. And once you can get industry interested, then other things open up, in terms of reimbursements, in terms of developing this. And this is, I think, the space where we want to go. Does that make sense? Okay, so to tie this part of the talk up in a bow, here's what this could look like. You have a cocktail of phages against Pseudomonas. You have a cocktail of phages against Staph aureus. You deliver these to the lung, and you get rid of just those two microbes. You're not actually getting rid of, all the good bacteria that live in our guts, that live in our mouths, that help us digest food. Nobody is going to be sad if you get rid of Pseudomonas aeruginosa in the body here. Similarly, Staphylococcus aureus, lives on some of our patients' skin, but it's mostly in the context of the human body, not doing anything particularly good or beneficial. And so if we can develop cocktails and potentially combine these with antibiotics, now you're really talking. So I'd like to kind of tie up this first part of the talk. Again, I've got two stories, one with phages causing problems, one where phages can be part of the solution, and offer that phages are a promising solution to the problem of antimicrobial-resistant lung infections. And that if we're going to do this, it's going to look something like these phage cocktails that can cover diverse strains of bacterial pathogens without necessarily hitting the good bacteria. And then finally, this approach of complementarity grouping that allows us to identify the phage receptors can be a powerful tool in informing the development of cocktails, and we're very excited about that. And I would offer that, through Nina and Kevin's work, this became possible and really through support from the CFRI. So thank you very much. Before I go on to the next part of this. Okay, so good phages and bad phages. You'll notice that these phages here that I'm pointing to look very different from the ones I showed you before. These filamentous phages are these long, thin things that look nothing like the lunar landers that I showed you before. And I'm going to argue that lysogenic phages like this one potentially cause disease. And with this, I want to get back to something I alluded to earlier, which is biofilms. And so any of us who have taken care of CF patients know what these look like. But essentially, these big, goopy, thick layers of polymers and bacteria that kind of can fill the lungs even today with the better therapies that we have. These are really problematic, both because they're sticky, they adhere to lungs and other surfaces, but also because they keep antibiotics in the immune system out. And because of these things, we end up using a lot of inhaled antibiotics. This isn't news to any of us. But how those antibiotics get in and how they work is really dictated in no small part due to the presence of, by the presence of these biofilms. So in terms of thinking about biofilms, this may not be intuitive, but dental plaque is a really great example of a biofilm that most of us have. And what it actually looks like, if you were to look at it under a microscope, is there on the left. It's essentially bacteria, but also the polymers that they live in. And the way I think about this is a little bit like bacterial biofilms being kind of like an anthill or a termite mound. On one level, you'd like to think that the unit of organism is the ant or the termite. But actually, if you wanted to get rid of the termites in your garage, you need to get a shovel and get rid of the entire termite mound. In other words, both the substrate, in this case, the dirt, But in the case of biofilms, the polymers, as well as the organism, the ants or the bacteria in this metaphor, they're one organism. And you have to think of biofilms in part as kind of an extension of the bacteria that make them. And so we're talking about CF lungs, but actually biofilms are a problem in most other chronic infection contexts. So if anybody in the room has ever had a urinary tract infection and had to take antibiotics for several days, or maybe somebody who's had a hip infection, with some prosthetic hardware or something like this. The reason why those are difficult to clear and you have to take antibiotics for 2 weeks or what have you is because antibiotics have trouble getting through biofilms, and the biofilms are crazy tenacious and difficult to clear. And that's, again, the reason for the long-term antibiotics and the chronic infections in our patients. And so here, the question that we started off asking was, how does Pseudomonas do it? How does it make biofilms? And so one of the first postdocs I had the pleasure and honor of working with was this guy here, Pat Secor. He's now got his own lab at the University of Montana, and he studies Pseudomonas and CF, amongst other things. And what Pat did is he added a polymer that's present in sputum and in wound secretions called hyaluronic acid to a solution of Pseudomonas. I'm not sure why it's so difficult to see there. But essentially, when he did this, he saw little structures like the ones inside that box that sort of formed spontaneously. And then when he added hyaluronidase, which is an enzyme that dissolves this polymer, they went away. And so he thought this was really cool because it suggested that Pseudomonas can grab polymers in its environment, sputum polymers, wound fluid polymers, and use them to build a house, to build a biofilm that protects it from antibiotics and the environment. Pretty cool. And it turns out that you don't actually need the Pseudomonas. You can just take the supernatant, the culture fluid, and add it to solutions of polymers, and it makes this film. It goes whoomp on a little glass cover slip. That's Pat taking a pipette tip and running it across the glass cover slip. And you can see it basically makes a furrow. And when we did atomic force microscopy, you can see it makes these sheets. And it turns out you don't actually need the supernatant. Pseudomonas is making this filamentous bacteriophage that is responsible for these structures that I'm showing you. Basically, it uses a virus to build a house as the bricks and mortar of its biofilm formation. And unlike the phages that I showed you a moment ago that swoop in and kill the bacteria, these are different. These are phages that the bacteria has domesticated, if you want to think about it this way. And these phages bud off the surface of the bacteria in huge amounts and don't actually kill the bacteria that produce them. If you take this phage and you mix it with different polymers, collagen is a polymer that's in our body, it basically spontaneously makes structures like this one. If you mix it with DNA, it makes structures like this. If you mix it with serum or urine, it makes structures like this. Basically, this phage that Pseudomonas makes in the context of any fluid in your body that has polymers will make a biofilm. How cool is that? So the question we wondered is, how does this work? And so one polymer that Pseudomonas makes in spades is this guy here. It's called alginate, and it's a big component of all the Pseudomonas biofilms. And you can see where on this slide, where Pat put the drop of phage. Essentially, almost immediately, it starts organizing the alginate into these networks, into these structures that are an important component of biofilms. And so I'm a real science fiction geek. Probably some of you guys are as well. Who's read this book, Cat's Cradle? Anybody? Okay, so in this book, it's by Kurt Vonnegut, and back in the, I think, 50s or 60s, I'm not sure exactly when, he wrote this story about a molecule called ICE9. Kurt Vonnegut's stuff is actually kind of scientific. It's science fiction, so it's fun. But ICE9 is a crystal, and anything that ICE9 touches turns into more ICE9. And so at the end of the book, the whole world has turned into a big thing of ICE9. And so the idea here is that crystals have this incredibly favorable state of entropy. And so when ICE9 touches other things, it's a little like pushing a rock up a hill. The rock wants to be at the bottom of a hill. And so in that same way, when ICE9 touches other things, they turn into more ICE9. They turn into crystals. And so the book is really about the Cold War and sort of mutual destruction. But it's also about physics and these ideas of entropy. And looking at these slides here, what this reminded me of is this book, Cat's Cradle, because not unlike that world that gets taken over by Ice-9, literally in seconds, this whole slide of polymer gets organized by this phage into a biofilm. And so what we wondered is maybe Pseudomonas is actually organizing CF sputum into a liquid crystal. So one property of crystals is that they're birefringent. They polarize light. Light can go through this way, but not through this way. And this is the sunglasses you probably wear when you ski. And so building on this, we asked, can we use polarized light to look to see whether or not Pseudomonas biofilms are actually crystals or crystalline in nature? And that's why they're so tough to disrupt. And so if you look at alginate under a polarizing light microscope, it's not birefringent. If you look at this phage, which is called PF4, it's not birefringent. But if you mix the two, all of a sudden you've got birefringence. That's pretty cool. And if you actually, titrate in the alginate and titrate in the phage, you get these phage change diagrams, a little bit like ice, water turning into ice. If you remember, graphs like this from high school, it's essentially a material moving from being a liquid into a crystal. And this is cool because it suggests that, this phage is actually organizing polymers, in this case, alginate, but again, hyaluronan and mucins and other things like this into a liquid crystal. And it's not just polymers. If you actually look at Pseudomonas colonies, this is a little bit difficult to see here on this microscope, but essentially small colony variants and other versions of Pseudomonas that make robust biofilms are crystalline. And that's really cool. And it's not just Pseudomonas, E. coli, Klebsiella, other gram-negative bugs that have big, robust biofilms also use filamentous phages like this one to build their house. So how does this work? What we think is actually going on is that the phages are kind of semi-rigid rods, and they're essentially these clouds of highly, anionic or negatively charged coat proteins. And if these things are free to rotate in space, they repel other polymers in the body that have similar charge. And it turns out that DNA and hyaluronan and alginate, all of these things are very anionic. And in dense solutions, what happens is, a little bit like if you and I each had a magnet and I were to push my magnet towards your magnet and these things would kind of repel each other, that's what's happening with the phages and the polymers. And provided you've got, enough cations there to mask those charges, you can actually assemble spontaneously a crystalline lattice. And it turns out that one of the things that characterizes the CF lung is an awful lot of ions, right? I mean, that's the whole point of the chloride channel. And so you get this hyperviscous, sputum that's chock full of ions and is completely ripe for organization like this. And let me show you what this actually looks like. This is one of my graduate students, Yolene Sweer, adding a drop of this phage into a solution of hyaluronan, which again is a polymer that you can find in sputum. And this is 3 minutes of footage. Okay. So what you're looking at is the organization of sputum polymers. We can do this with mucin and DNA and artificial sputum into a crystal. Essentially, the bacteria is using this phage to organize sputum polymers into something with higher order and with a very favorable state of entropy. And I would actually say, in a perverse kind of way, kind of beautiful. And what I would argue is that biofilms in the CF lung made by Pseudomonas and by these other bugs are tenacious and difficult to clear for the same reason that diamonds are forever. They have this incredibly favorable state of entropy that is difficult to disrupt. So what? Well, I would argue that this matters clinically. And so if you look at those biofilms that I mentioned before under scanning electron microscopy setup like this one here, PaO1, which is a standard lab strain of Pseudomonas, looks kind of gnarly and nothing like anything you'd want to have in your lungs. If you delete this phage, because again, it lives inside of the Pseudomonas genome, and we call that the Delta PF4 strain. Now you have this really wimpy biofilm. Those little Swiss cheese holes are actually the filter that we're growing this biofilm on. And you can see on the bottom, you can't see any filter, any Swiss cheese. But on the top, the biofilm is so wimpy that it's being washed away. And if you look at antibiotics, so tobramycin is an antibiotic that we give a lot of our patients in the lung. And if you increase the concentration of that and look at colony-forming units of bacteria, I mentioned rough strains don't make very good biofilms. Small colony variants, SCV, make big biofilms. The SVV isolates are really resistant to the Pseudomonas, or to the tobramycin rather. They'll grow until you get very high concentrations of antibiotic. The rough colony variants are susceptible. If you take a strain that lacks this phage, called PAO728, it looks like a rough colony variant. But then if you spike in this phage, now it behaves like a small colony variant. It acquires the ability to keep out antibiotics. If you then, that's a shame. If you fluorescently label the antibiotic, it gets caught in these structures. That's what you should have been able to see there. But what's more impressive are slides like this one. The red there in the middle is colonies of Pseudomonas that are growing. The green that surrounds it is a shell of this bacteriophage that the Pseudomonas is making to protect itself. Here's another version of this that hopefully you can see well. You have the Pseudomonas in the middle, and maybe not unlike an M&M or the little pigs hiding from the wolf, it essentially builds a shell of this phage all around it to keep the antibiotics and the immune system out. This is what this actually looks like on a functional basis. And so we developed an assay to look at this. We make these biofilms on the filters. I showed you this a minute ago. And we allow the antibiotics to go across, and we look at how that affects a different bacteria, here E. coli. And what we find is that some antibiotics, so as trianam, amikacin, tobramycin, are trapped by these networks, and other ones actually get through. And that'll be important in a second, as I'll show you. Then we asked in our CF patients, together with Carlos, does this matter clinically? I don't need to tell you about cystic fibrosis, except I do want to highlight that one of the things we look at with this is pulmonary lung function in 1 minute, FEV1, and antibiotic resistance. And what we find is that in a population of our CF patients, this is a small number of individuals, but if they're infected with Pseudomonas, they tend to have, I'll show you in a minute the numbers on this, or many of them tend to have this phage, and they have a lot of it. So in these sputum samples, they have as many as 10 to the 8. That's 1 with 8 zeros after it. Phages, just this phage per ml of sputum. A milliliter is about the size of my thumb. So that's 1 to the 10 to the 8 of this phage in just a little bit of sputum right there. It's incredibly abundant. And if you actually look at the sputum from our CF patients, if they're infected with this phage, it's crystalline. And so we measured this. We developed a polarizing light microscope that can quantify birefringence. We represent that as sign D. And essentially, sputum from our patients who are infected with Pseudomonas is crystalline. If they're not infected with Pseudomonas, it's not. And if you spike back in this phage, now your sputum is a liquid crystal. And so working with Carlos, this is really part of the project that he and Liz here really drove. We looked at whether this phage is abundant and prevalent in populations of patients. And what Liz and Carlos found is that essentially in a cohort of 58 CF patients at Stanford, this was found, this phage is present in 36% of these folks, so just under 40%. If we looked at 2 larger cohorts, one from Denmark of 474 patients, and another one of the Pseudomonas.com genome database, which at this point had 2,000 isolates in it, about 40% of the patients were infected with this phage. But what was really remarkable is that, over time, if you look at little kids, these pediatric patients, only a few of them had this phage. If you look at adults, it's about 50-50, but in a cohort of pre-transplant patients, they basically all had it. So there seems to be a selective advantage for, having this phage over time in our older patients. And then Carlos and Liz looked at essentially what this meant in terms of Leeds criteria, which, as many of you guys know, is kind of the standard we apply to understand whether people have chronic Pseudomonas infections. And if you were infected with this phage, you were much more likely to meet those criteria for chronic Pseudomonas infections than if you didn't. And then what was really interesting is when we looked at antibiotic resistance, Essentially, the same phages that got trapped in that in vitro assay I showed you a minute ago were also, the same antibiotics that got trapped by the phages in that in vitro assay were also the ones that, these individuals tended to have resistance to. And that allows you to put together a model like this one here, where if you're infected, if you're a patient with CF infected with Pseudomonas, aerosolized antibiotics, which are these little red dots here, can kind of get through this slimy mucus barrier and kill the bacteria that live there. On the other hand, if you're infected with this bad bacteriophage, and if Pseudomonas is producing it and organizing the sputum into a liquid crystal, now the antibiotics are trapped on the top and they can't get into the bacteria within. And you'll remember that kind of candy coating shell picture that I showed you a moment ago. And over time, this leads to more infections, worse resistance, and bigger problems. So to tie up this part of the conversation with a bow, I would offer that we can conclude a handful of things. The first is that Pseudomonas, a bacteria, partners with the bacteriophage to build biofilms. How dastardly is that? Then I would offer that this phage contributes to the pathogenic features of these biofilms by keeping out antibiotics, and I didn't show you all this data, but by making them more adherent and more robust. And then finally, these phages are associated with antibiotic resistance in CF Pseudomonas infections. I don't know if Carlos is going to tell you about this later when he gives his talk, but we're trying to think about ways to use screening tests for this phage to identify or to give some information about outcomes and antibiotic use and this kind of thing. So we think this is a really exciting frontier in terms of thinking about the microbiology of the CF lung and Pseudomonas infections in particular. I think I'm almost out of time, so I won't spend a lot of time on this. But the other thing that we've been doing is trying to develop vaccines actually against this filamentous phage. And so one of the fellows in our group, Christian de Vries, noticed that the coat protein of this phage is identical across a lot of different clinical isolates. This is 669 clinical isolates. And so based on that, we have a mouse wound model of infection. And so Christian immunized these mice. And we find the way we do this assay is we titrate the amount of bacteria in, so about half the mice get a wound infection. And what we did is we first immunized the animal against this coat protein called CoAB. We boosted them, and then we made these wounds and infected them with Pseudomonas. And then 3 days later, looked to see which mice were infected and which ones were not. And it turns out that if we immunize against this filamentous bacteriophage, we can prevent many of the mice from being chronically infected with Pseudomonas. This works even better if we come in with monoclonal antibodies. And this is really exciting. As I mentioned at the beginning of my talk, we're working with a company called Inimmune, which is a spinoff from GlaxoSmithKline, to develop this vaccine and have been working in pig models, and we have data with that. But what we actually think is going on here, maybe to cut to the chase here, is that ordinarily antibodies, which are what you elicit when you immunize somebody, are really terrible at sticking to goopy polysaccharides. They want to stick to proteins. They're really bad at sticking to carbohydrates and slimy layers. But because this phage, as I showed you a minute ago, decorates the surface of Pseudomonas colonies, now you've got protein. And so what we think is going on is that these antibodies, which we labeled here with little gold dots, essentially stick to the phage on the surface of the Pseudomonas, and now the immune system can engulf it, which is what this shows here, although I won't go through that data. The other interesting thing about these antibodies against this phage is that they allow antibiotics to get through. So we made 5 monoclonal antibodies. One of them didn't work very well. And when we look at the size of these bacterial colonies, if we added, before the colony grew up, this monoclonal antibody, all of a sudden the antibiotics can get through. So to tie up this whole talk in a bow, I'm going to offer 3 things. The first, and I'm going to go out of order here, is that lytic phages can help protect individuals with CF from bacterial lung infections. Conversely, some of these lysogenic phages can actually cause disease. Different phages matter. We need to understand this biology if we want to treat our CF patients effectively. And then finally, a vaccine against this particular phage may have benefit in potentially protecting our patients from cystic fibrosis. And then finally, I wanted to end on a note of thanks to the CFRI and to Siri and to all of you in this room for supporting this research and for filling the pipeline, not only therapeutically, but in terms of scientists who are going to work on these problems, both in academia and in the private sector. Nina has actually gone off to Gilead. She's working on antimicrobial therapies. And I think I chalk this up as a win and a success for CFRI and certainly for our lab. And then finally, just wanted to acknowledge our collaborators, including my friend Carlos right over there, but then also Pat Secor and Gina Suh and some of our other folks who've gone off and are doing their own things, their own labs. But it's great to be able to share all of their work with you today. And I thank you very much for your attention.